needs to be robust. And what we sometimes find is when we get to the end of a discussion with regulators, that it's enough to be significant for PFS, but there can be no indication of harm for overall survival. And usually where we end up is that the trial is significant for PFS, but that the observed hazard ratio for overall survival is less than 1. So if you have a significant PFS outcome, but your observed hazard ratio for OS is greater than 1, then there's always the possibility that although you're significant for PFS, you may actually be causing harm. So that can't be excluded. And that puts you into quite a difficult position. There are therapies now that are not expected to have an effect on the size of the tumor. So there are targeted therapies that just turn the tumor off and inactivate them, but just don't change the size of them. And then the RECIST criteria don't really help at all. And if that's the case, you wouldn't expect to have any effect on the size of the tumor. You may expect there not to be significance for PFS. And in which case, then you might want to be really only interested in being significant for overall survival. But again, you might want to exclude harm on PFS. This could also be due to late treatment effects and immunotherapy, which tends to not have a particularly good effect on PFS, but we do see late effects on overall survival a bit further on. So if you're in this situation, then you might not be expecting to see significant for PFS, but you would be expecting to see something for OS. And then the question then is, how do you manage the multiplicity between PFS and OS? So there are a couple of different ways that this is done. Typically, if we have PFS as the primary endpoint, there's a fixed sequence. So you will test PFS first, usually at the 5% level. And then later, when you're doing your final analysis for OS, you would test that for significance, but only if the PFS has been significant in the first place. So there's a fixed sequence of testing, and type 1 error is protected that way. If you're in a situation where you might not be significant for PFS, but you might be for OS, then you could split alpha between the two endpoints. So you could just do a bond for only split, not particularly efficient, but it is usually what's done. And so, for example, you might test PFS at 4%, leaving 1% for OS. And again, this can be pretty effective as used. What we usually see is a more complex type 1 error management, usually through some graphical multiple testing plan. You can see on the right here that there's a representation of a fallback procedure, actually, for moving your alpha around depending on what's significant. Okay, so it's important to manage endpoint multiplicity. So here's an example, a study designed for PFS, looking at the implication of OS, and I'll follow this example all the way through the talk. We're managing the multiplicity from the two endpoints through a fixed sequence. We require enough PFS events to detect an increase in the PFS median from 6 to 10 months, which is a hazard ratio of less than 6. Usual power and significance, and we have an interim analysis for futility at 50% of the information. And the futility assessment is based on conditional power, which is the conditional power at the interim. If that's less than 10%, then we'll call it futile and not proceed. Or if there's stunning efficacy, then we will stop, and hopefully the regulators will allow us the approval based on that. And then there's some more details about the sample size there. So in this example then, 180 PFS events from 250 patients. We can see on the right-hand side here that the recruitment and the event dynamics is really important in estimating when the interim analysis is going to take place. And we can see here that according to the plan, the interim analysis is going to be happening at 18 months, when we might expect the final analysis at around 30 months. And the timing of these interim analyses is really important. as we start talking about sample size re-estimation, for example, there's no point doing an interim analysis after all the patients have been randomized. And so you would want that to occur a few months before the planned end of recruitment. So finally here, let's talk about seamless phase 2/3 trials. I want to talk about two types of phase 2/3 trials. One is operationally seamless trials. So this is where we have a phase 2 study followed by a phase 3 study, usually at the same sites. And here then you'll have two separate protocols, and the phase 3 study will be powered independently of the phase 2. But the big advantage here is that the sponsor can review the phase 2 results in quite a lot of detail, and then perhaps adjust their plan for the phase 3 study moving forward. So operationally seamless, then you have a sample size in your phase 2 and an independently powered study for your phase 3. This is, if you like, normal phase 2/3 studies, just that it's operationally seamless and not inferentially seamless. What I'll be talking about is inferentially seamless studies, which is where we have phase 2 and 3 in the same protocol. This is really attractive because phase 2 patient results are used in the final comparison, and there is a saving to be had there. But there is a downside as well. The downside is that the sponsor cannot review the phase 2 results prior to starting the phase 3 part of the study. This is the job of the Independent Data Monitoring Committee. And the Independent Data Monitoring Committee will recommend actions to the sponsor based on their viewing of the data. We find a number of sponsors are very nervous about not seeing the results of their drugs study partway through, and this is something that they have to get used to doing. So it's the bottom one that I'll be talking about, which is the inferentially seamless studies. So adaptive trials in oncology is very commonplace now. Almost all trials in phase 1 and 2 are adaptive in nature. Phase 2 studies, we do find in some rare cancers that they can be pivotal, and when they are designed, they're mostly Bayesian or have Bayesian decision criteria in them. Single-arm pivotal studies are seen in this space. If we're in a more common cancer, then you're expected to do a randomized study, and adaptive phase 2/3 seamless confirmatory trials are now fairly well used. And there are some adaptations that now meet the criteria for adaptations with well-known characteristics, which is a regulatory term for they're usually allowed without too much discussion. And these are sample size reassessment and population enrichment studies. However, there are more complex adaptive designs, which you would then need to get into very early discussion with regulators, the complex innovative design initiative by the FDA and the Scientific Advice Working Party at EMA, for example. So if you have a synthetic control arm or you're looking at a Bayesian single arm, sorry about the typo there, then that's the route that you would take. So let's start by talking about sample size re-estimation as a potential for seamless phase 2/3 study. So this is where we've gone through the earlier phases. We have a strong expectation that this drug will work. We're in an area of cancer where the endpoints are extremely well known. There's no usual discussion about what the phase 3 is going to look like. So you already know approximately what you're going to do, and you're put in front of you the options for having separate phase 2 and phase 3 study, or doing a seamless phase 2/3 study. So in this space then, we have low information on some of the things that we would normally need to design a phase 3 study. And that is, we don't really know how good the treatment effect is, or the control group response. So the control group may be relatively new, or it may be that the control group responses are improving over time anyway, and we just don't know where we are. We do find that powering for the smallest clinically important effect is expensive. And if you understand that many biotechs survive on investment income, then they find that these types of designs are actually more attractive. Okay, so we'll certainly see here there's now quite a lot of use of promising zone designs, which is what these are called. And there's actually been a recent systematic review where they've actually summarized the reasons for using a sample size re-estimation design. And here are some of them. There's only 15 that they found, but mostly it's because they really don't know how good the treatment effect is going to be. So let's have a look at the plan. This is the diagram of the sample size re-estimation. So the study starts, and at an interim stage, we look at the interim results and we decide whether there's already efficacy, in which case we'll stop the study there and then, whether the results are currently favorable, in which case we'll go to the planned end of the study. If the results are promising, but we think the power to get there is a little low, in which case we may increase the size of the study and the number of events or the number of patients. And then we may go to an adaptively increased number of patients. Whether the study is currently unfavorable, in which case we'll go to the planned end but not hold out much hope, or whether the results are already futile and we can stop now. So this is a sample size plan. When we talk about these designs with clients, they start to say, well, really, if it's unfavorable, we just want to stop at the interim. Thank you very much. So actually that unfavorable design, part of the design just disappears, and we just have a larger futility zone there. So what happens is we get to the interim analysis and we start to do some calculations to see which zone we're in. And this is how it looks then. So to be in the efficacy zone, we should have already met the criteria for early stopping according to our chosen alpha spending rule. That puts us in the efficacy zone, and we can stop there already, provided we've had prior agreement with the regulators that stopping there is something that they would agree with. We have a futility zone, which is defined by conditional power. So if the conditional power is below, say, 10 or 15%, the DMC would recommend stopping for futility. If the conditional power is less than some minimum level, around 30, 40, or 50%, it may be called unfavorable. If the conditional power is between, say, 30, 40, or 50% and 90%, then that's the promising zone. And then we may increase the sample size to achieve a conditional power of 90%. Or if we're currently favorable, then continue to the planned sample size with confidence to do that. So this is really what SSR adaptation is about. It's about deciding which zone we're in at the interim analysis. The tricky bit is, if you're in the promising zone, is how many patients to increase the sample size by. And again, there's a whole raft of literature out there about doing that. What we need to do, though, at the end of the study, is if we've run this study, is that we need to produce a p-value at the end of the study that controls the type 1 error. And again, there is a methodology for doing that. This methodology has been around for a while. Kui, Hung, and Wang, who all worked at FDA when they did this, published a method that's now fairly common. And the way this works then is that the study is split up into 2 stages, those events that happened before the interim analysis and those that happened afterwards. And we generate 2 independent stage-wise test statistics, Z1 and Z2, and we combine them according to the planned weight of the number of events in the 2 stages. So for example, if we had 50% of the events before the interim analysis and a planned number of 50% of the events after the interim analysis, then W there will be 0.5. And that's how we would combine those independent test statistics to come to a valid test statistic at the end. If you imagine that we increase the sample size in the second stage, then you don't change the weight. The weight is the planned weight. And so it could be that you've doubled the sample size afterwards, and you could have, twice as many patients in stage 2 as are in stage 1, but you still have to use the planned weight. And then there's always a discussion about, well, aren't those patients then worth less in this analysis than those in the first part of the study? So that's usually the discussion point around this. So there is a valid way of dealing with the phase 2/3 part of the study. It's by finding a test statistic in stage 1 and a test statistic in stage 2 and combining them in this way. And this again is a fairly common way of combining test statistics from 2 stages, particularly in this and other adaptations. So let's look at our example. Remember, the example was a hazard ratio of 0.6, and here we're designing optimistically now. So we're setting a planned sample size of 190 subjects and 135 events. And we are planning the zones, as you see here. I've used p-values and conditional power. And on the right-hand side here, I've just put that in terms of what the observed hazard ratio would need to be in those zones. If we go into the promising zone, then we would then increase up to a maximum of 180 events, the exact number we would calculate, and potentially up to 250 patients to get that at the right time. Let's see how that works. There is a bit about performance here. If we look at the top row here, so this is the alternative hypothesis, where we would hope that the treatment gives us a 10-month median compared to 6 months. The interim zone probabilities are as you see. And you can see here then that in the promising zone, there is just over a 30% chance of being in there. And by increasing the number of events in that zone, we have a conditional power in there that would be much higher than it would be as if it was a GSD design. So if we have promising to favorable results, then this type of design is usually better than a GSD design because of the conditional power that we increase during that point, even though the unconditional power may be slightly less. Okay, let's move on to the population enrichment phase 2/3 design. So here then is where we have a known subgroup of patients in our trial population where we suspect that the drug is going to be very effective, but what we're hoping for is that the treatment could be effective in the full population. So we split the trial population into a subgroup and its complement. We want the flexibility to test the treatment in the subgroup if the treatment is not effective in the full population, in the complement of the subgroup. And this can be then if you've developed a drug that is aimed at a particular biomarker in a cancer, and you think, well, if it's going to work, it's going to work in that subgroup, but actually we suspect that it's going to work in the wider group of patients. And so we then have this plan for population enrichment. It's very similar to the previous plan, except there's a new zone. And the new zone we'll define for you in a minute. But in the new zone, the enrichment zone, this is where we switch from looking at all the population and we just look at the subgroup of interest. And there we may actually stop recruiting patients outside of that subgroup into the trial. So this changes the recruitment pattern for the trial too. And you can see that there's still the potential to increase the number of events, but there's also here the potential to enrich and change the testing from the full population to the subgroup population. So this is based on predictive enrichment. We divide patients into subgroups according to some predefined, well-validated biomarker. And we see the study will materialize into independent time cohorts. So the first cohort recruits from the full population, and then after the interim analysis, the second cohort recruits either the full population or only the subgroup population, depending on the first cohort of patients. So at the interim, we decide to continue with the full population, or to continue only with the subpopulation, or stop the trial for futility. So these are some of the decisions that we can make at the interim. Here are the interim zone definitions again. I've got an example of this, so it should be a bit more obvious when we get into the example. But essentially, we have a multiplicity feature to deal with now, because we have either the full population or the subpopulation that we can move forward with, and we have to adjust for that. So the final analysis is based on a combination of p-values. So we have a p-value from cohort 1 in the full population, p-value for the subpopulation in cohort 1, a p-value for the full population in cohort 2, and a p-value for the subgroup in cohort 2. And the question then is, how do we combine those to retain the type 1 error? Well, it's done by closed testing, and anyone involved in closed testing, this will be quite familiar with you. We look at the full population being significant only if both the full population hypothesis and the global null hypothesis are significant at the local level, and for the subpopulation, we test it's significant if the subpopulation hypothesis is significant and the global null hypothesis is significant too. We also have the 2 stages to deal with. So this is how the p-values are combined. So if there's been no enrichment and we're looking at the full population, then we can declare significance if both of those 2 combinations of those p-values be at the local level for significance. And you notice then that W1 and W2 appear here. They're the weights according to the events in those populations. In the case of enrichment, then the combination is just subtly changed, where we're starting now to use the subgroup only. Okay, so let's have a look at an example of that, just to show you how that looks. So again, here we have a PFS study where we have a first interim analysis where we have an early look for futility for PFS. We have a second interim analysis where we're looking for the first time for efficacy and adaptation and enrichment. And then we have a final analysis for PFS as well. And you can see here then that I've started to include overall survival in some of the simulations. So the population here is assumed to have an incidence of 60% of the population. Median PFS is 6 months, median OS is 24 months. And the size of the treatment effect you can see here is that for PFS we expect, or we hope, that the hazard ratio is 0.6. But we expect for overall survival that the hazard ratio is 0.8. So here's the design. I'll get over this fairly quickly, as we've just covered some of this. And here's the predefined adaptation plan. Now this looks quite busy, but it's the kind of level of detail that we need to get into to fully specify exactly what's going on. And this is the kind of detail that appears in the predefined adaptation plan that we put together. I've just put it in a table, but this is the kind of detail that we get down to. So you can see it's based on conditional power in the full population, conditional power in the subpopulation, and so on. So again, this is the kind of level of detail that we need to get into as we predefine these. Importantly, we tend to work out how well this trial is going to operate by thoroughly simulating the design. This involves simulating correlated PFS and OS times, we could look at a whole choice of all the different design parameters, different timings of the interim analysis, different choices for conditional power, different choices for the size of the interim zones, all the different stopping rules. This has to be robust to recruitment patterns, dropout rates, different types of treatment effect, different types of control group response, and really looking at the range of possible timings for interim and final analyses. When we do this, we tend to come up with some fairly detailed simulation plans. And again, here's an example of a simulation plan that we might put together for a trial like this. So you can see that for all the different design parameters, there's quite a big number of settings. When you multiply them all together, you're getting close to 1 million different combinations of these designs very quickly indeed. And so the question then is, how do you simulate all of these? Well, the answer is you don't. You do some sort of directed search through these and come close to what you think is a good design. There is some software now that's available where you can get at a large number of these different settings. So we use software that does this for us. You see on the right-hand side, there's a heat map there of the different scenarios and the different designs. And the density of the color there is proportional to a performance criteria that we have that is based on speed, cost, numbers of patients, and power. And you can see on the left here that we can do lots of different summaries of all the different designs. So each design there occurs as a dot on that scatter plot here. And through here then, we come through to find what we hope to be a close to optimal trial design. So for you, I've picked out 3 of the scenarios. The first one is the scenario where the treatment works as planned. The second one is where the treatment only works in the subgroup and doesn't work in the complement. And scenario 3 is where it just doesn't work at all anyway. And so these are, a couple of good scenarios to look at. And thoroughly simulating all of these to understand the operating characteristics is really important. So if the drug works as expected, then we can see on the left-hand side here, there's a strong chance that the study will already be stopping for efficacy at the interim. And the reason for that is it was chosen to be relatively late. And you can see on the right-hand side that most of the studies will stop around the 24 months time, and they will stop at the second interim analysis for efficacy. There are some studies that go further than that. There are some studies that go into the promising zone. And this gives a feel for when the study will stop and the different decisions that are going to be made. I've done these for the other 2 scenarios as well, so you can get a feel for how these look different. Scenario 2 then, you can see the treatment only works in the subgroup, and you can see now that there is moving into the promising zone, and the enrichment zone is now much more frequent, and stopping for efficacy there means that usually you've got a longer study on your hands if you haven't already stopped for efficacy. Scenario 3, if you like, is the null scenario. And you can see here now many of these simulations stopping it into analysis 1 or 2 for futility, or even going into the unfavorable zone there and not being successful. So through here, we get a feel for how well the design works. One of the things we can also get out of simulation is the estimate of the treatment benefit. And these can be pretty important. So for scenario 1 here, I've plotted out the observed hazard ratio for PFS and the observed hazard ratio for OS at the time of the final analysis for PFS, but only for those trials that were significant for PFS. So you can see on the left-hand side then that to be significant for PFS, observing a hazard ratio of less than about 0.8, 0.78 is what's needed. And you can see that if you're stopping for efficacy at that interim analysis, you're likely to have a biased outcome. You're likely to have a biased estimate because we know the truth here. The truth is 0.6. But if you've stopped at that analysis, you're likely to be averaging at a hazard ratio of around the sort of 0.55, 0.56 mark. So that means then that we do see bias, and we see bias in estimation all the time in adaptive studies. On the right-hand side, you'll see the distribution of the observed hazard ratio for overall survival. And one of the things that we look out for when we're doing these simulations is what's the chance of having significance for PFS, but having an observed hazard ratio for overall survival of more than 1. So what we do is we look to assess the probability of that happening by chance, and looking to, one, know what that is, quantify it, but also to minimize it. If the successful outcome of the trial is to be significant for PFS and have an observed hazard ratio for overall survival less than 1, anything in that grey zone then would not be a successful study. So we have to be sure that we're on top of quantifying that. So I've mentioned about estimation after adaptation. Estimation after adaptation is known to be biased. Traditional maximum likelihood estimates tend to be biased because of some selection at the interim analysis. And early stopping affects the sampling distribution of the estimator, as we've just seen. So point estimates and confidence intervals need to be bias adjusted. What we've seen over the early part of the publications around adaptive design is an almost total focus on p-values, adjusting p-values, keeping type 1 error under control, and not quite so much emphasis on the estimation. But more recently now, there's been many more publications and work and research on estimation after adaptation. And so now you'll see much more use of adjusted p-values, median unbiased estimators, repeated confidence intervals, and resampling methods to get good estimation after adaptation. For SSR, we do see that the level of bias is usually quite low, as is the reporting of bias-adjusted estimates in the publications. Actually, that's very low too. And for population enrichment, again, the bias is quite low there as well. But again, we hardly ever see any reports of bias-adjusted estimates. And I would imagine that over the next years, this is one of the big developments that we'll see, which is reporting of these adjusted estimates. Operationally speaking, I've talked about pre-definition and thorough simulation of all of these. So you need to have a very good simulation capability, and you need to be able to predefine the adaptations to a level that is adequate for full definition. We've, in the group, have been working with FDA and EMA for the last years on fully specifying these. We're pretty used to what it is they need to see, but we do see that every now and then they ask for a little bit more to be part of our standard reporting. So it's quite interesting how this is still a moving feast with those. The biggest question that sponsors usually get around adaptations is around minimizing operational bias. Here, independence of the data monitoring committee, firewalling the results from the study teams, and usually the whole sponsor, independent stat teams for interims, very thorough DMC charters, and written trial integrity documents so that you document exactly how you're going to firewall the results from the teams are all common and expected now. Operationally, it's not enough now just to open the database and start cleaning it near the end. You have to have an early database construction and real-time data entry and cleaning as you go so that you're able to have confidence in some of the decisions that you're making at the interim analyses, particularly around adaptations. You wouldn't want to find out that key data have changed after the adaptation that would have changed your decision. So operationally, there's implications for these studies as well. With these types of designs, from the perspective that I have, we see that adaptive phase 2-3 designs are routinely accepted by regulators. There's guidance out there now from FDA and EMA, and we're about to see ICH E20 appear, which will be an ICH document about adaptation and adaptive designs. Whichever design we look at, early regulator interaction and agreement on the design is absolutely essential. That's usually done in Europe through a scientific advice process, and at FDA through Type C meetings and so on. And with other regulators, it's good to not surprise them with an adaptive design at the end, but to get buy-off on the design. We share adaptive design documentation with the regulators to demonstrate predefinition and operating characteristics, and expect to submit your synopses and simulation plans and adaptation plans very early in that process. Okay, so last slide then, and finished with a few minutes spare. There's now a lot of experience designing and documenting adaptive trials, and best practice is now being determined with many of these things that you'll recognize from the talk. Thoroughly simulating the design, documenting simulation performance, documenting in the predefined way the decision rules in an adaptation plan. Regular control of type 1 error is now routine. Minimizing operational bias, and make sure you have early interaction with those regulators. So thank you very much for listening, and I think we now have a few minutes for questions if there are any, so I'll hand back to Julie.
Thank you so much, James, for your insights and sharing all your experience over gosh, well over a decade. There's one question in the chat at the moment, and that's asking what software was used for the simulations.
Okay, so there are different bits of software available. There are some software available in R. GSD, RPact are good R packages. We're lucky in Cytel that we have our own software, so we use East to do the simulations and the calculations, and we have a new cloud-based engine for supercharged East. It's called Solara, that allows us to compare hundreds of thousands of trial designs together. In fact, some of those heat maps were from Solara, so we do have those. But yeah, there's a lot of R packages out there that can be used to help you design these phase 2-3 studies.
Thanks for that. We just have time for one more question. Liz, would you like to ask it in person?
Yeah, sorry. I was just interested in the alpha splitting approach. So you mentioned giving one of the endpoints 0.04 and the other 0.01. Do you give more alpha to the endpoint that you think will have the bigger treatment effect?
Yeah, that's an interesting discussion, because at the time you're doing the analysis, you will have an idea of how many events that you think you need and that you'll get. So, for example, if you're essentially sizing for PFS, you just look at the implication on OS, and it may be that you don't have many OS events, particularly if there's a long time between median PFS and median OS. So it comes down to how many events you think you'll see. And it also comes down to what you think your treatment effect might be. It might be that you suspect no effect on PFS and everything on OS, in which case you may have it the other way around. I've seen alpha splitting being done equally in favor of PFS and in favor of OS. So there's no real sort of rule for this. The only advice I would give is try and have an extremely good understanding of how many different types of events you'll see at different analyses, so that you can predefine accordingly.
Thank you.
So that's perfectly timed answer. So many thanks, James, for a great talk today, and best wishes for everyone in their designs.